This lesson is going to be about DNA mutations. So the first type of mutation that we're going to talk about are insertions and deletions, which are point mutations where a single base pair is either added or removed. So if we look at this example, we have our normal sequence with our DNA listed on top, and then the complementary RNA below, and then the amino acids that correspond with that RNA sequence. And so if we look at an example of an insertion mutation, let's draw our DNA strand. And if we're going to insert a base pair, let's just throw in an adenine right here. And then we copy the rest of the sequence as is. And so then when we make our complementary RNA based off of this new sequence, we're going to get AUG. Now, we'll denote this as our mutation. We're going to get UCG, and then we're going to have UAC, and then these extras, we're just not going to worry about those for right now. So here's the amino acids that we're going to get from this sequence. Amino acids. So if we compare this back to our normal sequence, we see that we still get the same first amino acid, obviously, because this wasn't mutated. But then our next amino acid, our second and our second and our third amino acids are different than in our original unmutated sequence. So we can see how this changes everything just by adding a single base pair. So now if we look at a deletion, if we look at our DNA, let's write our sequence. Let's just go ahead and remove this G from here. There's our deletion, and then here's the rest of the sequence. So, oops, when we base pair this, and then we have our amino acid again, we get this as a result. So we see a similar result to the insertion in that our amino acid sequence that we're getting is going to be completely different, and it's going to change everything. So mutated sequence denoted here and here. So these are what we like to call frame shift mutations. And this is because even though we're just adding in a single base pair or removing a single base pair, it shifts our reading frame with our 3 base pairs over, which changes everything and changes your amino acids. So that's how that type of mutation works. The next type that we're going to talk about is substitution, where in this case we're going to exchange one base pair for another. So here we're not doing a frame shift mutation like we were in the other one. Our frames are still the same, but we're still going to get a change. So in that specific amino acid, I guess, is what I'm trying to say. So if we look at our sequence again, we have our DNA, and let's go ahead and in our second reading frame. Let's change this C to a T. And then the rest will just be the same. So we got rid of the C, we brought in this T. That's where our change happened. And so we write our RNA and then our amino acids. Again, the first one's the same because we didn't change anything. Second one, we have a change here. So now we have a different amino acid. The third one, again, since we didn't shift the reading frame this time and we didn't change anything here, this one's going to be the same. But this is still a mutated sequence. And so this is how we can see the difference. The top ones, although we're only making a single change, it shifts the reading frame to the right one, which throws off every consecutive amino acid after the mutation. Whereas a substitution mutation, since we're not shifting our reading frame because what we're removing we're replacing with something else, our reading frame remains the same, so only that specific codon will get switched and result in a different amino acid. The subsequent ones coming after the mutation will still remain the same as the normal sequence. And so these genes get passed to all the cells, and sometimes the mutations can be beneficial. So sometimes they can be beneficial, other times, a lot of times, they're going to be harmful, or they can have no effect. And the reason for this is because it depends completely on where the mutation is occurring and what proteins are going to be involved and what the functions of those proteins are. So it's completely different, and that's why we have such a wide spread of effects of mutations.